It was like twice where she was like looking for a calculator like during lab, and I was like, I feel like this her. She kept leaving her stuff very much. Thank you.
Guys, our class has gotten smaller. What's going on?
Oh, yeah.
All right, so we're gonna finish up where we were. Oh, first of all, did you guys who are in my lab section, did you get my email? So there's a video to watch. Try, it's only 4 minutes long, and it helps, it really helps because it shows you the technique. We'll go over it in lab today, but it shows the technique of how to inject the sample. That's kind of a difficult thing to do. All right, so last time we left off, we ended up with talking about the Nobel Prize winning work of Barbara McClintock and her transposons, also known as jumping genes. So that was a big revolution. But you figured out the corn plants did this, they have jumping genes. That leads to some of these beautiful corn that you see here, but we also know that humans also have that now. So jumping genes are fairly widespread. All right, we're going to move. So that's one source. The point of that was that's one source of mutation. So the jumping gene could jump into a gene and completely disrupt it. It could actually jump into a place where it would actually cause an increased expression of a gene. It could also, again, jump into a gene or somehow disrupt it and stop the expression of a gene. But there are other types of disruptions that can occur. One is what's called a point mutation. This is a change in a single DNA nucleotide. And there are different types of point mutations. So one type is what's called a base substitution. So we talked about the bases, right? The nitrogenous bases. So in a base substitution, you have one DNA nucleotide that's replaced by another. So for example, you might have an adenine that's replaced by a guanine or something like that, right? So it's just completely changed. And there's no other change, right? But that's enough to then change the protein sequence, right? It'll end up swapping one amino acid out for another, and that can have really large impacts on the resulting protein. So here are the possible outcomes. So one possibility is there's no effect at all. And you might ask, well, how is that possible? How can you not have an effect even though you swapped out one amino acid for another potentially when you have a base substitution? Well, let's go back a second. Can anyone think of why that might be? Well, how can you change essentially a base, which would change the codon, right? And yet not have any change. Let's go back to look at we had a very important chart we had earlier. If you look here, it gives us an indication, right? There's 64, when you have this triplet code, there's 64 possibilities, which we worked out mathematically the other day. But you only have how many proteins make up your body? Not 64. How many proteins do you have in your body make up your cell? 20, right? 20. Okay, and that's the same pretty much for all organisms. It's about 20. All right, so here's a possibility. Look, UUA, that's the codon, the code, right? That gives us leucine. So those three amino acids code for leucine, the amino acid leucine. But what if the A is swapped out for G? You still have UUG, right? But guess what? You still get the same amino acid, right? So that's a situation where you can have a mutation and yet it would have absolutely no effect whatsoever, right? So that's referred, and there's a bunch of other possibilities here, right? Like you can see here. Let's do another one. Here's valine down here. GUU, so guanine uracil uracil. Well, you can have guanine uracil cytosine, and you still have valine, right? So it wouldn't make any change, even though you swapped out one base for another. Let's get back to where we were. So that's would be called it is a mutation, right? You've changed the genetic code, but there's no effect at all. So those happen pretty often, actually. So what about some other possibilities? So it may produce a completely different protein. So that's a substitution mutation. So a good example of that is in sickle cell anemia. So we have in sickle cell anemia, which occurs in places where there's malaria. So it's fairly common, and what happens is with that is you have a change in the protein that's found in red blood cells, right? And that red blood, so this protein is normally there's 4 subunits to make this protein, and it carries oxygen, right? It's called hemoglobin. Now, when you have this substitution, you still make the protein, but it's an oddly shaped protein. And so within the red blood cells, the proteins now sort of clump together. They stack together, which they shouldn't do. And they're no longer as soluble. They're normally just sort of floating around. They're soluble within the red blood cells, even though they're fairly large proteins made of 4 subunits. But now they sort of stack together, and this changes the whole shape of the red blood cell from being this characteristic round disc-shaped red blood cell to more of a sickle shape, right? So that's why it's called sickle cell anemia. Anemia means there's not enough oxygen and nutrients getting to the tissues, to the blood. So that's an example of a substitution mutation in which just a single base, there's a single base substitution, and yet that completely change. What happens is it changes the amino acid sequence. So one amino acid is swapped out for another amino acid, and that completely changes the structure of the protein. Go ahead.
Does sickle cell anemia ever occur in Caucasians?
Yes.
It does.
Yeah, so it's not, people have this misconception that it only occurs in Africa. It certainly occurs in Africa. There's parts of Africa where there's a high level of malaria, which of course is carried by mosquitoes. But there are other places, I believe in I can't remember exactly. I think in certain parts of, like, I think maybe Greece or some other places in the world where it has nothing to do with Africa. It's completely not in Africa at all. There just happens to be a lot of malaria there. And the thing about sickle cell anemia is if you're heterozygous, it's actually protected. You can't really get malaria and die of it. Whereas, so there's a reason why the gene persists. It's actually adaptive. It's only when you're homozygous that you have a real problem with it. A heterozygous person may occasionally have some episodes during their life when they're not completely without any symptoms, but they're generally going to be fine. They're pretty much fine. All right, so that's a substitution mutation, so that can really affect things. And then finally, there's a frameshift mutation. So we're going to talk about the frameshift mutation I'm going to talk about in a second here. Let's take a look at the 3 possibilities. Here's the neutral mutation. Here, T is substituted for C. So if we look at this figure here, here's the normal sequence here. For example, the messenger RNA of AAU. Here in the original DNA, we have AAT. And so in the normal situation, you would have ASN, which is asparagine, so one of the 20 amino acids. And so that's the normal sequence. Now, what happens if you have, instead of AAT, you have AAC? Well, then you have a different messenger RNA. Instead of AAU, you're going to have AAC here, and that's going to give you. Hold on. Oh, this is going to be the same thing. It's a neutral. It's going to be a neutral mutation, right? So AAU and AAC and the messenger RNA will both give you asparagine. So there's no difference whatsoever. And so really there's a mutation, but there's no effect. So that's called a neutral mutation. And actually all of us have neutral mutations. There's plenty of neutral mutations out there. Because if you think about it, there's really nothing to stop these things from happening, right? If you had one of your ancestors had a neutral mutation, even in a really critical gene, well, it makes no difference, right? It's going to make absolutely no difference. So you probably have kept it, right? So your family has kept it. So every single human out there, including you, you have a bunch of these neutral mutations, and it's not really a problem. Okay, so here's the other type I mentioned earlier, which is a substitution mutation. Here's the normal sequence, CTA, and then if you look at the messenger RNA of GAU, and then here we have aspartic acid. ASP is for aspartic acid. You get a functional protein. You can see it has its active site here. Here's the mutated sequence. We have instead of CTA, we have CCA. And if you look at the messenger RNA, it's GGU instead of GAU. And now instead of aspartic acid, you have glutamic acid. Okay, so remember, each amino acid is unique in that they have an R group, also called the side chain, and those are very different, right? Some are charged negatively, some are charged positively, some are not charged, some are polar, some are non-polar, and so forth, right? So changing out the identity of the amino acid can have a really big effect on the three-dimensional structure of the resulting protein. So in this case, we have a completely non-functional protein. It doesn't even have this sort of groove that's typical of a lot of proteins that are enzymes, right? So it doesn't have an active site, and so it's not going to do anything. Okay, so this could potentially be fatal. Sometimes people can have this sort of mutation, but biochemically their cells work, they might have a workaround, but often this is fatal, right? So when you have these sorts of mutations in really important enzymes, important for like basic, cellular processes, that individual basically never makes it beyond maybe a few cells worth of embryonic development, right? So a lot of potential living organisms just simply die very early on, right? Okay, and the example I gave you earlier was with hemoglobin, which is a protein which carries oxygen. And that's what they're mentioning here. Sickle cell disease is due to a base substitution. There's essentially a change at one base, and that leads to a substitution where you have a new amino acid, and then the whole protein is now malformed. So here's the example here. So we have, each hemoglobin molecule. So here we're showing the actual red blood cells. Here's a normal red blood cell. It's roundish but it's disc shaped, and it's actually called biconcave because it actually is concave on both sides, right? The top and the bottom. And this allows these cells to squeeze through. You actually have some capillaries in your body that are smaller in diameter than a red blood cell. And so you can imagine the red blood cell just folding just a little bit to squeeze through. Okay, so that's what it should do. Here's the sickle cell anemia cell. It's more rigid. It's not able to fold away, and obviously it's not as smooth looking, right? It has these sharp edges that could sort of catch on things. So here's the substitution. So again, each hemoglobin molecule, so there's millions and millions of hemoglobin molecules that are floating around inside an individual red blood cell. Each one has 40 subunits. Each subunit has, I'm going to guess, around 400 amino acids, somewhere in that order. And here we're just showing a very short part of it, right? So we're just looking at this. In this one section, you have proline, I can't remember if this is glutamic acid. I think this might be GLUs, might be glutamic acid. And then here we have a mutant one where we have one base that's been substituted out. So instead of CTT, we have CAT. And now this glutamic acid is substituted in terms of amino acid. We're going to get a valine. If you look at this code here in the messenger RNA, GUA, it's GUA instead of GAA. That's going to code for valine. Okay, and that completely changes the structure of the protein, and the proteins clump together, and then we have the sickle-shaped cell. All right. So let's take a look at how a frame shift mutation occurs. So this is where you have one or more nucleotides that are either inserted or deleted from the DNA. And this is really a problem because it can completely change the sequence of codons. So here's an example, just using the English language. We have the cat ate the rat, okay? So that's a kind of a sentence that makes perfectly good sense, right? But what if we And so obviously what they're going, using this example where every word has 3 letters in it, just to make it similar to the codons, right, within DNA. If you remove the C, what do you get? Well, we're gonna keep reading with this. You can think of a sort of a frame being placed down here. So the first one's actually okay. The is fine. But we remove C, so now that becomes ATA is the next thing to read. So yeah, so the ATA, tet, her, at, right? Completely nonsense, right? So that's what happens when you essentially add a base into the DNA. But the reading mechanism expects every 3 should be a codon, right? But now it's complete nonsense that it's reading. What if you add a C? So this can also happen, right? So the CCA instead of cat, so you can see how this has now shifted everything. So again, this is complete nonsense. The first 3 are fine, but after that everything's bad. The first 3 letters are fine. So the first codon, so you get the first. If this were coding for amino acids, the very first one would be correct, but all the rest of them would be incorrect. So let's look at this here. So frame shift mutation is the addition or deletion of a base. Here's an addition here, an additional T is added next to it. So here we have, let's look at this in detail. Here we've got ATA, and then if you look at the messenger RNA of AAU, that's going to code for ASN. I think that's asparagine. It's been a while since I've memorized all my. I had to in biochemistry. You have to learn all. You have to memorize and know how to draw every single amino acid. Okay, so it's been a while since I took biochemistry, but so there's an amino acid there, and then everything's looking fine. Here's glutamic acid. I think here, and now we have threonine, asparagine, and then what happens here is we've added what have we added? We've added AAT, so AAT. Now we're adding an extra T. You see that here? We've added, and then this GAA after that. We added extra T here. And then instead of glutamic acid here, you're going to now get the next one here, which is this codon here, which is instead of GAA, we're going to get, I wish they lined this up a little bit better. GU. It's flipped over. Yeah, I guess it would be UGA, and that's going to be a stop codon. Okay. So remember, let's go back to what is a stop codon? You guys remember what a stop codon is? So if you look at this chart, which is really helpful, so there are 64 possibilities with this, codon triplet code. We have 3 different possibilities that will actually stop the production of a protein, right? Remember that we talked about, you saw the animation, the thing comes in and it stops the production, and then that ends the reading of the sequence of codons. So UAA, UGA, and UAG. So what's happening here is we've hit a stop codon. And that just stops, so that stops the protein from being made, right? Within the ribosome. So then you have this really short bit of protein. So a polypeptide that's kind of short, it's not going to fold properly into the functional protein. And therefore, that protein, which might have been an enzyme, is not missing from the cells. Okay. So why is this stuff important? Well, a lot of you might be interested in health issues and things like that. It's a big thing for us, in terms of, life and so forth, economy and so forth. So in the US, the 3 deadliest forms of cancer are lung, colorectal, and breast cancer. Okay. And there are other ones like prostate cancer in men. It's very, very common, for example. But does anyone know there's breast cancer month? Does anyone know when that is?
October.
It's October. Okay. Do you know when prostate cancer month is? it's November. So it strikes me as kind of weird, like everyone knows it's October for breast cancer month and there's all this stuff. And then, you know, prostate cancer, which only affects men, of course, right? Because only men have prostate glands. It comes around in November and it's just like silent. No one cares. Anyway, so here's the development of cancer involves a series of accumulating mutations that can be different for each type of cancer. So there's the most common types of cancer are due to mutations of two types of genes. The first are tumor suppressor genes, okay? So what are tumor suppressor genes? Well, these are genes that specifically help to control the growth of tumors, okay? So unfortunately, what can happen is that you can have mutations and you can have growth, you know, unusual growths of cells, right? And this leads to cancer and so forth. But the good thing is you actually have genes that are already there on hand to stop this, right? I don't know if you remember, but we talked about the cell cycle. Remember that? And there are certain control points in the cell cycle where the cell essentially takes stock of what's going on. Has DNA been replicated correctly? Has the DNA been split up equitably between the two daughter cells from the mother cell? There's all these sort of points in which the cell takes a look to see if everything's okay. So these are what's doing this. Well, it's actually these tumor suppressor genes. They're looking for problems with replication of DNA, all sorts of things, right? And then if that is the case, then it will essentially do various things. It can try and correct the errors, right? And if that's not possible, it can then essentially suggest the cell kill itself, which is apoptosis, right? Which is a controlled cell death, and that gets rid of the cell that had the faulty DNA, right? So that's what tumor suppressor genes do. They normally are stopping you from having tumors. But what happens with certain types of cancers is those tumor suppressor genes themselves become mutated, right? And they no longer function. So now that nothing's holding back these cells now that have the faulty DNA, and they'll start to replicate and in an uncontrolled manner. Okay, so that's why, does that make sense? Why? So they're kind of like the guards or the policemen. You can think of them in the cell. And if you take them out and remove them, they're no longer working, they're faulty, then the cancer can start and continue to grow, right? So tumors, by the way, like almost everyone, and it's probably gonna be shocking to you, like they've done studies where they take people that died of something else at like a very ripe old age, like, you know, way into their 80s or 90s or something. And they've taken people like that and they've like carefully looked, you know, they donate their body to science. They're like, hey, go ahead, go, you know, do some science with our body. They go through and they section the body, and then what they find is there are all these little weird growths that have occurred, kind of like moles would be an example of that. But even like internally where something started to grow, but then it just stopped, right? What stopped it? Well, it's these genes, these tumor suppressor genes will just hold things in check, okay? All right, so another type of gene that is often mutated, and it's found a lot in the press, people talk about these, are proto-oncogenes, okay? So an oncogene is a gene that's actually going to cause cancer, okay? Proto means before, so it's a gene it's one that, but it's before, it could potentially cause cancer if it becomes mutated, okay? So it seems like nonsensical. Why would you have cancer-producing genes in your body, within your genome? Well, you don't really. What these proto-oncogenes actually have a very important function, which is to stimulate cell growth. So imagine, you know, you're on your bike and you fall and you scrape some skin off your knee or whatever, right? You want certain genes to be activated that tell your cells to start to replicate like crazy, right? To make new skin cells and so forth, repair the capillaries, you know, all the stuff, right? So you want those genes around, but so there are specific genes that will suddenly become very active in those sorts of situations and will help to repair your body. But the problem is, so they're very stimulatory. They're like, think of them as like the cheerleaders. So like, okay, you know, we got damage, but don't worry, you know, we're going to start building some more new skin cells and some more capillaries, and we'll do all these things, right? But what happens when those cells get mutated, right? Now they're constantly activated, and they're just telling their cell, you know, essentially the cells that they're in, to just keep replicating over and over and over again, right? Even though there's no need for it, right? It does, even though it's completely nonsensical to do that. So that's why, so basically, these are the major types of genes that are affected that lead to cancer. Okay. So yeah, these are, so what do they code for? These genes actually code for proteins. The codes and the proteins are going to turn specific genes on or off by binding to nearby DNA. So that's getting more into what these genes actually code for are transcription factors. So again, these are the transcription factors of proteins that go back. They're built out in the cytoplasm. They move back through the nuclear pores into the nucleus, and then they actually bind to the DNA, and they can tell other genes to turn on or other genes to turn off. So that's how they functionally work. Okay, the tumor suppressor genes and the proto-oncogenes. Yeah, so let me see. So I think this is just what I've talked about already. The proto-oncogene is a gene involved in normal cell growth. So again, normally they're stimulating your cells to grow. That's a good thing usually, as long as it's done right. Mutations or changes in the proto-oncogene cause it to become an oncogene, which causes the growth of cancer cells. Yeah, so again, when it says these, this is referring to the proto-oncogenes, or the proto-oncogenes may control DNA replication, cell growth, and cell division processes. So those are all growth processes, right? So the proto-oncogenes are involved in growth processes. The tumor suppressor genes are more like, they're inhibitory in the sense that they're saying, hey, let's check out this DNA, make sure it's okay. Okay, so carcinogenesis will begin. So this term carcinogenesis sounds very complicated, but just like to generate, genesis means to generate something, it's generating cancer. So the beginning of the generation of cancer is caused by the loss of the tumor suppressor gene because it becomes non-functional, or so the loss of that gene, or it could be a gain of the proto-oncogene activity. So it could be an increase in this proto-oncogene activity because it's not really a proto-oncogene anymore. It's now an oncogene because it's been mutated. As a result, cell division occurs uncontrollably. And so here is just a reminder of the sorts of things we looked at before. So in the typical cell cycle, you have two major phases: interphase and then mitosis, right? So mitosis is the M phase. So we have various checkpoints, like we have the G1 checkpoint. This is the main cell checkpoint. If DNA is damaged, apoptosis may occur, and it would be the tumor suppressor genes controlling that. Otherwise, the cell is committed to divide when growth signals are present and nutrients are available. So now it's going to go on. We have the S phase where DNA is replicated. S stands for synthesis. And then the G actually stands for gap, but there's a lot going on. They're essentially growth phases. So G2 phase is growth and final preparations for division. Remember, we're going to need a lot of microtubules for making the spindle fibers and so forth. Okay, so the G2 checkpoint, this is a mitosis checkpoint. Mitosis will occur if DNA is replicated properly. So again, typically you'd have something like a tumor suppressor gene helping with checking out whether the DNA has been replicated properly. Apoptosis may occur if DNA is damaged. So again, who's controlling that? The tumor suppressor gene. And then finally, we have the M checkpoint. Remember, as you go from prophase to prometaphase and metaphase, where everything's lined up. Then anaphase, where the DNA is split to the two daughters, to the two poles of the cell. Well, right here at metaphase, there's actually a checkpoint there. There's a spindle assembly checkpoint. Most mitosis will not continue if chromosomes are not properly aligned. Okay, so it's tumor suppressor genes that inhibit cell division at the G sub 1, G sub 2, or M phase control point. And so this figure really pertains mostly to the tumor suppressor genes, but then we also have the proto-oncogenes. They turn on or maintain cell division. So they're kind of the cheerleaders in a sense. They're saying, hey, it's time to divide, that sort of thing, right? They're cheering the cell on to go ahead and divide. So the proto-oncogenes become oncogenes, and then they're very problematic. Go ahead.
Maybe more of a metaphysical question.
Right.
Is there any level of decision making going on there? Do you have a consciousness?
So that's an interesting question. So normally your cells play together really well, right? So individually, cells will even voluntarily die for you, right? So is that consciousness? I don't think so. It's probably pre-programmed, right? But all these things are programmed. So for example, if you have a cell that's been infected by viruses, it will actually put signals on its cell surface indicating to your immune system, please come destroy me, right? And then your white blood cells will come and destroy that cell so that the virus can't replicate. So most of these cells are sort of self-sacrificing, right? They're doing things for you, right? That's the only thing that makes sense in evolutionary sense, right? That you whatever you are, you're a collection of trillions of cells, but somehow there is this idea, probably through evolution, that the only way this is going to work is if everyone cooperates, and that even means cells dying, you know. So, but I like your idea, because what if a cell decides one day, wakes up and it's like, you know, I don't want to die. I want to replicate, right? I want to go crazy and make millions and millions of copies of myself. That's kind of what cancer cells do, right? But ultimately, that then kills the individual. So then they die with the individual. So it's not a very good strategy. But anyway, you could write a science fiction plan, a cell that didn't want to die and doesn't want to cooperate. That's what cancer cells are essentially, right? So mutation in either of these two classes of genes leads to uncontrolled cell growth or cancer. All right. So what are some specific genes you may have heard of? I think these you might have heard of in the bottom. We'll get to those in a second. So let's start with P53 though. This is a major tumor suppressor gene that's frequently mutated in human cancers, more than any other known gene. It acts as a transcription factor. Remember what a transcription factor is? It's a protein that's made in the cytoplasm and comes back through the nuclear pore into the nucleus, and it controls other genes. And these other genes are ones that will inhibit the cell cycle. So we'll slow down the cell cycle. They may even stop it. They may even prompt, some of these inhibitors genes will actually prompt apoptosis, which is programmed cell death. So that's when the cell neatly sort of decides to die. So it blubs up. I showed a picture of this in a previous lecture. So P53 is kind of a master gene in a sense, right? It controls all these other genes, right? So that's why if you specifically have a problem with P53, then that can lead to cancer. So you can have mutations in some of these other genes. Like imagine there's like 6 genes that are controlling cell inhibition of cell cycle. If you knock out one, well, there's still 5 other genes, right? So it's maybe not as big a deal. But when you knock out this master control gene, then that leads to cancer. Okay. So P53 is really significant, comes up a lot when we talk about cancer. There's another one that's really significant too. This is the RB gene, which codes for retinoblastoma protein. It's a tumor suppressor gene, right? So mutation in this gene can lead to a retinoblastoma, which is cancer of the eye that affects the retina, okay? So that's pretty terrible. You can get cancer in the retina. I had a friend that actually had that. And they had to put a little the doctors put a little radioactive pellet inside his eye to kill that region of the retina. It did work because he survived. So a lot of these things are pretty terrible and they're very common. So this is a gene that can come up a lot. Now the next one is also very common. This is the BRCA1 and BRCA2. This functions also as a tumor suppressor gene. Mutation of these genes can lead to breast cancer. So it's very common, right? One of the most common types of cancer. Ovarian cancer, another very common cancer in females. And it does actually cause breast cancer in males, and it actually causes prostate cancer in males, okay? So this made the news a number of years ago. Does anyone remember? No? You remember? Yeah, so there's the famous actress Angelina Jolie. She did a test to see what genes she had. In fact, she had one of the two. I can't remember which one. And so she elected to have some surgery, pretty aggressive surgery, I think, to remove some breast tissue and so forth, because her mother had actually died of breast cancer. So it made sense. Anyway, so these are very common genes that can cause cancer and anyone really. So here's what happens. Let's look at normal P53. We have ionizing radiation. This could be ultraviolet light, right? So put on, wear your broad-brimmed hat in the summer, put on the sunscreen, your nose, the ears, so forth, right? ionizing radiation could be also be X-rays, things like that. You don't want to get additional X-rays beyond what's really necessary. This causes DNA damage. Induction of P53 causes cell cycle arrest. Oh, so normally this P53 it's a suppressor, right? So it causes cell cycle arrest, prevents the cells from replicating. Now, if you have damage to this gene, if the damage is really extensive, this could lead to apoptosis. So this is what normally happens. Yeah. So if you have DNA damage, this gene turns on, and it tells the cell, look, your DNA is too damaged, and we're going to go to apoptosis to save the human. Or again, P53 is a master gene. It's turning on a bunch of other genes. Some of them can do gene repair, right? So they're going to go in, repair the DNA and fix it, and then we're off to the races, right? Everything's fine. We're going to keep going. Here's what happens when you have a mutant P53. So you have ionizing radiation. It causes some damage to the DNA. But this cell, it's already a tumor cell. It's lacking, the P53 has already been mutated by a previous ionizing radiation or something like that. So there's no cell arrest. So it could be that the damage is so terrible that the cell will just simply go through necrosis, meaning that it can't live because it's been so damaged. So you know, mitotic failure and cell death. So that's actually kind of a good thing if the cell dies. But here's the bad thing. What if the damage is just bad enough that the cell now starts to go through uncontrolled replication? It's multiplying, dividing, but it's actually healthy enough they can continue to do that. Well, this now leads to cancer. So this cell is now dividing at a high rate, and each time it's dividing, because it's already damaged, it has a higher rate of mutation. So internally within this cancer cell, the cell that's going to lead to cancer, it's going to have even more mutations. And eventually what you'll have is a kind of evolution within your own body, within the tumor, where the most aggressive cancer cells, the ones that divide the quickest, and the ones that are able to free themselves from the tumor cell mass, those are going to be selected for. And then this leads to cancer, and the cancer can essentially spread throughout the whole body. Okay, very depressing topic, but essentially what's happening when you have a cancer is you have a kind of a evolution that occurs very quickly within these cancer cells in a very negative manner, right? Because now they'll replicate even faster. So mutant P53 can no longer recognize damaged DNA in an effective way, and therefore can't stop cell division. That's what's happening here. Cells continue to divide uncontrollably, and they form tumors. Okay, let's look at how this actually progresses. So although cancers vary greatly, they almost always follow this common multi-step progression. So most cancers begin as an abnormal cell growth that's benign, okay? So let's use an example of an epithelial cell. So this is a cell, let's say it's on your face. I'm gonna use your face because why? Because the face gets a lot of ionizing radiation, UV light, right, in the summer. And so if you look at any individual, like even someone as young as you, and people that if they take samples, like a little biopsy, taking little samples, you'd find cells that have mutated genes in your face. Why the face? Because this is exposed the most, right, to sun. Okay. Does that mean you have cancer? No, it doesn't. But you might get some odd growths, right? So this is a benign tumor initially. So it's the primary tumor. So we're going to code those as brown. So these are not necessarily all that dangerous yet, right? They're just, I mean, almost everyone, particularly if you get older, you have little bumps and things that have to be removed for the dermatologist, right? That's not cancer, but it's sort of a precancerous in a sense, right? Something to be paying attention to. Now, what happens is, well, you've got this growth, and one cell out of this bunch of cells that are they've already been mutated a little bit, like maybe one gene that's been changed. Maybe it's more ionizing radiation, or perhaps it's just chance, right? Because again, these cells are a little bit modified, they're not normal cells. They're dividing more than they should, and you've got another one that's mutated here. This one's much more aggressive. This one's going to divide at a much higher rate. So suddenly this thing that you've had for 10 years and it's just a little bump, didn't really change. Now it starts to really get big and change color. I mean, you should have probably seen the dermatologist already, but that's when you make an emergency, call the dermatologist and say, hey, this is really concerning. And then they get you in like really as quick as possible. Just find any dermatologist, because they have to typically remove this thing off your skin and do a biopsy and see what's going on here. If you don't do that, then what will happen is it will grow and grow and grow. And again, initially these are skin cells, so they should stay confined to the skin. But what if it's a mutated cancer cell? It doesn't know that it should stay confined anymore. It's mutated, right? The genes are changed. It starts to invade local tissue, local nearby, and it can either go into blood vessels or invade into lymphatic vessels. Either way, you're now able to circulate. And so again, I'm using an example of skin cancer. It could be other types of cancer, but then this would then go throughout the body. It could go to the lymph nodes. You can get cancer developing the lymph nodes. It could really go just about anywhere. It could go to the liver. It could go all these places in your body, and you can in your brain, and you can now have these cancer cells growing throughout the body. So, yeah. So here it's growing. It's coming out. It's gone through the lymphatic system, and now it's starting to divide into in a lymph node or somewhere else in the body, right? So most cancers begin as abnormal growth that's called benign. It's not cancer. It usually does not grow larger. A mole is actually an example of this, right? Okay. So don't get too concerned about moles, but something to just monitor and make sure they don't start mutating. Okay. Additional mutations may occur, and the growth may become malignant, meaning that it's cancerous and possesses the ability to spread. So that's what's essentially happening with this purple cell. Okay. So what's the take-home message here? Like, basically, we need to be a little bit vigilant. We need to monitor our bodies, go to the doctor. For example, colonoscopies. Forget the age now. I think they dropped it. I think your first colonoscopy was at 50 traditionally, but now it's reduced down to 45. So I think people were exposed to a lot more environmental hazards perhaps now. There's more plastics and things out in the environment. There's also people don't exercise as much as they used to and things like that. So there's who knows really why, but people are getting more colon cancer. So colon is the large intestine, right?
Isn't it supposed to be like 10 years before, like if it's genetic, like if a family member had it, you're supposed to get a colonoscopy?
Yeah, if you have a family member that actually had colon cancer, then you're at increased risk, right? So then you might start very early, right? You start doing it every 5 years from age 25 or something. So typically, colon cancer increases, the probability increases with age. But why am I suggesting that someone put a camera in your private part of your body? Because they do, they send a camera in there and look around. Why? Why do you want to do that? Why would you say? Because if you catch these little things called polyps in the colon early, they just snip them off, and that's it. We're done, right? You're good for another 5 or 10 years, right? But if you let that thing grow and it starts to mutate, then it turns into cancer, right? What about cervical cancer? You have to heard of a pap smear, right? So you're getting literally doing kind of a biopsy because you're getting some cells that slough off from the cervix. They look at that. If everything's okay, it's great. If there are some mutated cells, that's concerning. And then there's ways to go into the cervix. There's like surgery. There's also some methods where they go and they freeze the sort of damaged cells, right? And this sort of slough off and so forth. So it's literally like life and death, right? So what you want to do is make sure you go see the doctor, do all these checks on the right appropriate times, right? And you have to be knowledgeable about this yourself, right? Also looking at your skin. It's actually recommended, believe it or not, you should go see a dermatologist every year, right? It sounds crazy, but you get a good dermatologist, you go see them every year. They check the little bumps and things on your face and make sure none of them turn cancer, okay? And there's a bunch of other tests that could go on and on, like you check the breast for the females, checking the testicles for the males and things like that, right? So if you catch these things early, it can literally be just, okay, you go in, they go in, they snip the little polyp off, they snip the mole off, you're done, right? And you're cancer free. If you let it go, you can have cancer. All right, sorry, that's my public service announcement for making sure you get to the doctor, okay? On a regular basis and do these sort of checks and so forth. So cancer cells are genetically unstable. When a cell becomes cancerous, it starts to behaving abnormally and becomes more aggressive to invade other healthy tissues, okay? So cancer cells, again, they're like cells that have gone bad, right? They've gone wild. They do not correctly regulate their cell cycle. So cancer cells break all cell cycle control mechanisms that we talked about earlier. They can escape the signals for death. They simply don't want to die, right? This really damaged DNA cell normally initiates apoptosis. That's what it should happen. But however, cancer cells don't respond to the internal signals for apoptosis or program cell death signals. So they survive and proliferate elsewhere in the body. So Actually, one thing I didn't mention here is normally your cells are bound together. There's sort of an extracellular matrix and things like that to hold the cells in place. In cancer cells, we don't know why exactly, but we know that some of the genes that hold things in place, those are actually disrupted, okay? So now the cells aren't held in place very much and they can escape, okay? So the cells can survive and proliferate also in the body. When metastasized, cancer cells travel through the blood and lymphatic vessels and establish secondary cancer in other parts of the body. So this is another thing why you want to keep checking yourself. I mean, nothing's perfect, right? But you don't want your cancer to metastasize, right? If you catch it, say you do get cancer in the skin, right? I'll keep using that example, but you get it removed and you caught it before it metastasized, meaning before it spread throughout the body, then you have a very high probability of surviving, very high. But once it's metastasized, then it's gone to the liver, it's gone to the lymph nodes, it's gone to the brain. It's very difficult at that point. All right, so that's that for that chapter. I know it's very depressing, but the positive thing I think is just to make sure that you're staying healthy, trying to see the doctor on a regular basis, and also to eating healthy and exercise. Those actually, believe it or not, things like exercise are actually help you with lowering your cancer rate. Eating healthy food, I think, is also a good thing. And the book actually talks a lot about that. It has a huge section where a whole page where it just talks about the healthy choices you can make. Any questions on that? All right. Cancer is not a fun topic, no. It's terrible, but it's better to sort of confront it and like sort of realize what it is. And the other thing about cancer, I'm going to talk. Sorry, I'm sort of on a roll talking about cancer. But one thing that I hate, that I hear a lot, is when people blame cancer victims, right? So like they say, okay, if you had made the right choice, I just told you a big spiel about how making the right choices, going to see the doctor, exercising, eating healthy, all those things are good. Don't drink too much alcohol, do not smoke cigarettes, right? But there's plenty of people that get cancer that actually did everything right. They do everything right and still get cancer. Why is that? Why could you get cancer while you didn't do anything wrong? Does anyone know? Go ahead.
Genetics?
Yeah, it's genetics. Partly, it may be from part of your family, it might be in the genes. So that's a good point. But the other thing is, a lot of these cancers simply arise not because of ionizing radiation, not because of chemicals or anything. It's just because the DNA replication process itself can have some problems, right? There is a 1 in a billion base, as you go along the bases, right? You'll have a substitution error in 1 in a billion, just as the cells are dividing, right? And also there's certain things like reactive oxygen species, which I talked about earlier, that are just a part of life, right? So you can mitigate those risks of reactive oxygen, because basically as your mitochondria are working, they create reactive oxygen species. That's just a fact of life. That's how your body operates. Can you help out a little bit? Yes, you can eat. So dark green vegetables have certain vitamins that are antioxidants. Blueberries, fruits, things like that have vitamin C. That's an antioxidant, right? So eating healthy will help a little bit. But no matter what, these reactive oxygen species are occurring. DNA replication errors will occur. And so you can do everything right and still get cancer, okay? It's not, and actually one in 3 people will get cancer, okay? All right. Sorry, but because people are like, oh, you didn't do everything right. I hate that. Like, no, you could do everything right and still get cancer. It's not, don't blame, people tend to like blame people that are cancer victims, right? Oh, you got lung cancer, that means you must have smoked. And these people didn't smoke at all, right? It doesn't work that way. All right, sorry. DNA technology. So we're going to go on to a new topic. So this is actually some real, kind of an amazing chapter. It's chapter 26. So it talks about all the amazing technology we have now, and this is really growing exponentially. It's getting more and more interesting technology every day. So knowledge of DNA has led to the ability to manipulate genes of organisms. Cloned genes are now used to alter the genome of viruses or cells. This practice called genetic engineering has many uses, forming a product to treat cancer and genetic disorders. So there's actually lots of promising medical possibilities that are coming out of this sort of gene technology. So let's talk about cloning. That's always interesting, right? So What is cloning? It's essentially the production of identical copies of an organism through asexual means. So some organisms just naturally do cloning all the time. If you take a single bacterium and put it in a petri dish, it will divide into two, and those two will become four, those four will become eight, and so forth, right? Essentially, those are all identical twins, right? They're all the same. Now, do humans ever do cloning? Yes, there's cloning already exists, because if you have a zygote that at some early stage divides into two embryos, then you have two identical twins, right? Which we all know, we probably even know some identical twins, right? They're technically actually clones. They have the same genetic material, right? So those are that is a type of clone. But when we talk about cloning, we're often talking about gene cloning. There's another, so this is cloning of the whole organism, but there's also gene cloning. The term cloning is also used as gene cloning, but it's very similar in sense. It's the production of many identical copies of a single gene. So you could, for example, as a scientist or a doctor or something, you might be interested in introducing a gene into a bacterium, and you want that bacterium to, say, make insulin, right? But you really want that bacterium to make a lot of insulin. So what you do is you introduce the gene over and over again. So you might give 50 copies of the gene to the bacterium, then put it in the petri dish, and then it will make a tremendous amount of insulin. So that's a form of gene cloning, when you're replicating genes within something like a bacterium. Okay. So what are the uses of gene cloning? You can produce large quantities of genes, protein product. An example was, insulin or something like that, or some other medicine. Learn how a clone gene codes for. So in terms of understanding how these genes work, you can clone a gene and then look at the protein it makes. That gives you some information on what the gene does. Alter the phenotypes of organisms in a beneficial way. So that's what's going on a lot in agriculture now. They're modifying organisms in various ways, and what they're doing is essentially producing what are called transgenic organisms. So trans means sort of you'd have to look up the dictionary, but it essentially means like a cross. Like if you went on a trans-American tour, that means you're going across the US, right? That sort of thing, right? So transgenic means you're taking genetic material from one organism and putting it into another. So an example would be if you took a human gene that codes for insulin and put it in a bacterium, you've made a transgenic organism, right? Because you've taken a human gene and put it into a foreign place, which is the human gene, I guess, would be foreign to the bacterium. So it goes into the bacterium. Now you have a transgenic bacterium. Now there's also gene therapy. This is where you take clone genes and use them to modify human. Okay, we're going to talk about that in more detail later. So right now we're just doing a lot of terminology. So transgenic is when you take a gene that's foreign to an organism and put it in the organism. Another term that is recombinant DNA or rDNA. This is DNA from two or more sources. So we actually saw that this happens all the time in humans, just in normal reproduction. Remember we had the chromosomes, and you have a chromosome from your mother. You have 23 pairs of chromosomes, right? If you look at chromosome number 1 in one of your cells, one comes from your father, one came from your mother, right? But if you were to pass those genes on, there's actually a possibility that those genes can get mixed up a bit. How did that happen? Does anyone know? How do you mix up? How do genes from the mother get onto the chromosome from the father, essentially? How does that happen? It's a low probability, but around 1 to 3% of the time you get this crossing over event, right? Right. So during meiosis, there's the possibility of having crossing over from one chromosome to another. So that mixes up the genes, right? Between your mother and your father, and then you can make those are then passed on into your gametes into the next generation. So that's actually a recombinant DNA, because you've gotten two sources, the mother and the father. You combine them, you mix them up, right? It's recombinant DNA. So humans have been doing that naturally for eons, right? But recombinant simply means DNA from two or more different sources. They're put together into a new piece of DNA. So to make recombinant DNA, you need a vector. So what is a vector? A vector is something that is going to carry something, right? And so in this case, it's going to be a piece of DNA that foreign DNA can be attached to. So let's look at here. We have plasmids. Okay, do you guys remember what a plasmid is? Does anyone recall what a plasmid is? We actually did plasmid. We worked with plasmids in the lab the other day. Exactly. So if you look at a bacterium, here's your standard bacterium. I'll make a capsule looking one. You have your regular DNA, which is a big, it's usually a single loop, but sometimes there's multiple loops, right? It can be 2 or 3 gigantic loops. Well, relatively big loops, not as big as like a human chromosome. So that's the main DNA. And then you have your typical, you might have a whole bunch of little plasmids, right? So these are little mini loops. So those are plasmids, and they can actually be transferred from one bacterium to another, okay? And what do bacteria use it for? Well, they can confer things like antibiotic resistance from one bacterium to another. So there is, although bacteria replicate asexually, a mother cell will make 2 daughter cells or are identical. They can actually transfer DNA horizontally to other bacteria through plasmids. Now, the interesting thing about plasmids, they have their own replication mechanism. So when this mother cell makes 2 daughter cells, it has to replicate its plasmids also, okay? So these are accessory rings of DNA in bacteria. They're commonly used as vectors. So by vector, it means we're going to carry another gene. So we'll see pictures of this, but like what we could do is take one of these plasmids and we could break it open, right? Break the ring open. We'll discuss how that occurs. And we'll put in, let's say, a gene for antibiotic resistance. That's actually recombinant DNA, right? You take in a foreign gene, you put it into this vector, and then you can take this and inject it. Well, let's use this because I got a picture here. You can get it, you can actually get this bacterium to take up, you can make it so that the little pores that occur in the bacterium, and then this vector can, this plasmid with the new gene for antibiotic resistance ends up inside here, right? And not every, when you do this, typically not every bacterium will take it up, like maybe one in 1,000, 1 in a million or something will take it up. But those are then up, if you apply bacterium to your petri dish, these ones with the new gene are able to replicate and survive. Okay. Go on. So how do we do this? So how do we actually make this recombinant DNA? There are 2 basic enzymes we need to make recombinant DNA. The first is what's called a restriction enzyme. What does this do? It's going to cleave the vector DNA. So, normally bacteria have these restriction enzymes because what they're called restriction enzymes because they restrict the ability of viruses to inhabit and destroy bacteria. So normally, bacteria are constantly under attack by viruses. How do they? And some of these viruses have DNA rather than RNA. So they have these so bacteria naturally have hundreds of these restriction enzymes that will chop off the viral DNA. Okay, so it's essentially a defense mechanism. It's a primitive immune system used by bacteria to restrict the growth of invading viruses by cutting up the viral DNA. But it turns out these restriction enzymes are really helpful. They can be used in cloning as molecular scissors that cut DNA at precise sequences. Let's actually look at a picture here. Yeah, okay. Here's a good picture. We have some human DNA. We have a plasmid, which is going to be the vector. It's going to carry our gene. We get that from the bacterium. Here's the human DNA. We're interested in the insulin gene because we want to make insulin in the dish, basically. So we're going to use a restriction enzyme to cleave the DNA. But we're actually going to use the same restriction enzyme in both places. We'll use, again, we have like 100 restriction enzymes to choose from, so we'll pick carefully and find one that works well for us. We're going to use the same restriction enzyme to cut the plasmid vector. It's going to open up, as we're going to use to cut this insulin gene out. And then what's going to happen is these ends, the picture here? Yeah. So when you use a restriction enzyme, it looks for a very specific sequence here. Like I think it's here, it's GATT C, something like that. You see there, it's going to go and looks for that sequence, and it cuts at that very specific place. And then typically it'll leave you with these what are called sticky ends. So it doesn't just cleave straight down the middle, although some might, but this one doesn't. And it's leaving the sticky ends. So it cuts one end short, the other one long. So you have these four bases, nitrogenous bases that are exposed here. And over here, you have four bases that are exposed over here. That's referred to as a sticky end. Why is it a sticky end? Because the reason is it's called a sticky end because it's possible for another piece of DNA to come in and stick to this, right? As long as it has a complementary sequence, right? It should be able to stick to that. Let's go back to this picture here. So you can actually see it's a little, you have to squint a little because this picture is a little small, but they've actually showing this that you have the sticky ends here, where the two strands are a little bit different lengths. Same thing over here. So you use the same restriction enzyme for both cutting the human DNA as well as the plasmid from the bacterium. And so they're naturally going to want to, they can naturally bind to each other with their sticky ends. And then there's another enzyme called DNA ligase that seals the insulin gene into the plasmid. So now we have a recombinant DNA because we have DNA in the plasmid from the bacterium and DNA from the humans. That's the definition of recombinant. We're taking two sources of DNA, put it the other one DNA piece. Go ahead.
How does it specifically know which part of the DNA to cut to get these, like the insulin producing part out?
Oh, yeah. So I think that, yeah, there's probably a lot more work that's shown here, right? You probably would get, I'm not a molecular biologist, but you probably have to get the exact piece of DNA here, right? You don't want all the chromosomes because that sequence, for example, the GA, whatever sequence I told you, that's probably going to occur quite a few times, right? So you probably have to restrict yourself to one chromosome. So you have to somehow get that one chromosome by itself. And then you have to, again, maybe choose very carefully, like which restriction enzyme you're going to use, right? But I don't know all the details. So obviously there's a lot more to this than what's being shown here. But the basic principle you get, right? Which is you're taking the DNA, you're cutting it with a specific restriction enzyme. You use the exact same restriction enzyme here, but your sticky ends will be able to seal to each other. Okay. Okay, so the host then, so now we need to stimulate, we have certain techniques. We essentially use things like heat shock. There's a variety of different techniques to make this bacterium more permeable to these plasmids. One is there's a heat shock, there's osmotic shock, there's also a new technique, well, it's not that new, but called electroporation, where you use electrical current to make the membrane more permeable. Anyway, there's multiple ways to make so that encourages the bacteria to pick up the plasmid, okay? Now, not all will, right? It might be 1 in 1,000 actually picks it up, but you've got millions and millions of bacteria in a dish, right? Petri dish. So as long as a few pick it up, then you're good. And you might have some sort of like, you might also additionally have added a gene with resistance to some sort of antibiotic, right? So then you apply the antibiotic, all the cells that didn't pick up the plasmid, they'll all die, right? And now you just got your human gene and the antibiotic resistance gene, and of course you've got the bacterial gene too. But the point is now these bacteria have essentially been taken over. They have this human gene and then now in the form of the plasmid, and they're all going to start making insulin, and they're actually making human insulin, right? So in the early days, with treatment for diabetes mellitus, we actually used all sorts of insulin, like bovine insulin from cows, dog insulin, all sorts of stuff in the very early days, right? But that insulin is not exactly the same as human insulin, right? So with this genetic technique, you can literally give people human insulin, right? The exact same protein as human insulin. All right, so, okay, so we talked about this. So the restriction enzyme access, we can think of those as molecular scissors that kind of cut DNA in precise sequences. Then you have the DNA ligase, which will seal the foreign DNA into the opening, and the vector DNA created by the restriction enzyme. Okay. So restriction enzymes cut double-stranded DNA at a specific site. Just checking the time here. Okay, so I want to run over here. Right, sorry. All right, so now we've got, okay, here's an example. The restriction enzyme EcoR1 always recognizes and cuts DNA at the sequence GAT2C. After cutting, a gap is created in the pieces of foreign DNA the gap is created in which pieces of foreign DNA can be placed if there is a complementary occurring. Foreign DNA and vector DNA are cleaved with the same restriction enzyme. Single-stranded complementary ends of the two DNA molecules are sticky ends because they can bind to each other by complementary base pairing. Okay, so we talked about that initially already. All right, so then we're on to the next. So anyway, that's very important type of technology that was really revolutionary and continuing to be used. And actually that is easy enough that we actually do some variations of what we just did. We often do those in lab at the undergrad level. So we're on to now the DNA sequencing. DNA sequencing is a procedure that determines the order of nucleotides in a segment of DNA. So this helps to identify specific alleles and sequences. It can help facilitate development of disease treatments and reveal evolutionary history and relationships. So initially, like many years ago, DNA sequencing was extremely difficult, tedious process in which the DNA would be chopped up into small little bits. And then tags, like radial labeled tags would be added and things like that. It was extremely difficult, very, very long process. But now the technology has proceeded just at light speed, essentially. I mean, a number of years ago, they had this thing called the Human Genome Project, in which they were sequencing. The goal was just to sequence the DNA of a single person, and it took, I don't know, do you remember, James? It was like a huge process of like 5 to 10 years or something to do this. But it was kind of crazy. It was like as they were doing this project, it's sort of like being in an airplane and you're building the airplane as it's flying. They kept increasing, like part of the project was actually developing new technology, right? So they kept making the machines better and faster and more accurate and so forth as the project was getting nearer and nearer to completion. So it was one of the few gigantic projects. It was literally like going to the moon or something like that. It was that scale of project, that scale of money. It was really gigantic. But they actually came in under budget because the technology progressed so much. They actually finished a little bit early and under budget, if I remember correctly. But now the thing is that still cost, I don't know, like millions, if not billions of dollars, right, for that first sequence. But now, if you wanted to, you could get your DNA sequenced for like under $1,000 or something. I haven't checked lately, but you could. It's incredible. Like you just give them a DNA sample, and they're like, okay, here's your sequence. So, and that just keeps getting faster and faster. So the technology is just, again, progressed at such a fast pace, it's even hard to comprehend. So why is this useful? Again, if you wanted to know if you have a certain gene, like you're Angelina Jolie and your mother died of breast cancer, maybe you want to know specifically which gene you have. We can do that now, right? You can do that. You can look for genes and there's different levels. There's one where you sequence the whole genome, right? That's pretty laborious. Or you can just go in and look for very specific genes, right? Which is probably what she did. So that has progressed tremendously. Why is this important? Well, the other use for this is in forensic biology. So it's one of the main techniques used in forensic biology to find the perpetrator of a crime, right? What you can find is sometimes you can find semen. You don't need a lot. You can find skin cells. You can find a hair follicle, which will have DNA in it. So literally almost anything, right? Just very, very small bits of biological samples is enough to get a full DNA sequence from a person. Now, we don't typically do the full DNA sequence, and we'll talk about that. There's kind of a shortcut, which we'll talk about, that's used in forensic biology. The basic idea nowadays with these modern machines that are so incredibly amazingly accurate and fast is that what they do is they'll have dyes, fluorescent dyes. It used to be radio labeled dyes. Now they use fluorescent dyes. They attach them to the nucleotides, and they can show the order of the nucleotides using this automated sequencer. So it actually will tag these things, and they'll go through, and it'll excite it with a laser, and then it has a detector, and it'll see what color is coming off. So it'll just read one nucleotide after another, and it'll read millions and millions of nucleotides very quickly. Sometimes to do this, you may not have a lot of DNA to begin with, so you may need to make copies of your DNA segment or the gene, or perhaps you're trying to do the whole genome. You might make copies of the gene of the DNA to begin with. And so the way you're gonna do this, if you're let's imagine, for example, we're not trying to sequence the whole genome anymore. We're interested in a particular gene, right? We're going to use polymerase chain reaction to do that. So let's look at this technique. So polymerase chain reaction, or PCR, can create billions of copies of a segment of DNA in test tubes within hours. So it's going to only amplify specific targeted DNA sequence. We'll talk about how it does that. The targeted sequence is usually just a few hundred base pairs in length. And DNA, it uses an enzyme called DNA polymerase, as well as we're going to need raw materials to make the DNA. That's going to be DNA nucleotides, which you can just buy now. You just order a bunch of nucleotides and they'll come in the mail to you, right? And actually everything you order in these little kits now, it's very easy and simple now. The three basic steps that occur repeatedly, usually about, and you basically, it takes about 35 to 40 cycles to take a very small quantity of DNA and then amplify it by millions of times, billions of times. Okay. So how do we do this? So the first step in PCR reaction is to denature the DNA. So we talked about denaturing proteins before, right? Essentially altering the nature of the molecule. So what that means for DNA is you're going to normally DNA. I'm not going to draw it as a helix, but it's normally like this, right? It's a double strand, right? When you denature something, it'll come apart, and you'll have single-stranded DNA. Okay, so we're going from double-stranded to single-stranded DNA. Now that's less stable, but there's a reason we need to do that. Because the reason is because we need to get at the code within the DNA. So we heat the DNA to 95 degrees Celsius. Remember, 100 Celsius is boiling at sea level. So that's gonna, if you're at near boiling temperature, right? Then what you're gonna do is you're gonna add a primer. Okay. What is a primer? Well, what you do is you go to your companies and online, you say, okay, look, I want a primer that's, A, T, T, C, G, something like some sequence, right? This is going to be called our primer. And it's going to be complementary to this DNA. So it's going to come in here and it's going to find the complementary sequence on one of the strands of the DNA. That's our primer. And why do we need that? Because we know that our gene of interest is downstream. This is our gene of interest here. It's downstream. This is the gene we want to amplify. It's downstream of this primer, amplify. Okay. And then what happens is, so annealing usually between 50 to 60 degrees Celsius to allow the binding of a primer to the end of each DNA strand. So you get the primer occurring here. And now we need to do extension. So we're going to have, so this occurs at 72 degrees Celsius. So this thing is cycling back and forth in temperature very precisely. You've got all this stuff already assembled. You've got all these, the primers, the nucleotides, DNA polymerase, the original DNA snippets. They're all in this little tube, this little tiny test tube, right? Not a test tube, a little teeny micro tube. So now you're going to have this polymerase it's going to come in, and it's going to add complementary bases to each of the single DNA strands, creating a double-stranded DNA strand. So you have the primer. Here's your gene of interest. So you essentially, you've already started this here, but now you're gonna come in here with your DNA polymerase. It's gonna work, and let's imagine it's going in this direction. It's gonna work and work and work, and it's gonna make this piece of DNA, right? And then the next thing happens is so now you have a new piece of DNA bound here, right? Oh, now we're out of time, but basically what you're gonna do is you're gonna go, I'll stop here, but we go back to this process here. And now here's this original long piece. We've made this other shorter piece here that contains our gene, right? So now we have 2 copies of that. Go through another cycle, 2 become 4, 4 become 8, and so forth. Okay, I'll stop there. And we'll pick it up here at PCR next time. Oh, by the way, I have to make the homework, the quiz and the homework, I'll make available. I forgot to do that. I'll make that available to Blackboard today, okay?
Is this on the exam?
Yeah,
Okay.
we'll see how far we get with this, but I think we should be able to get through most of this chapter. It's a very interesting chapter, okay? It's not, yeah.